Welcome to the Deep Dive, where we take your stack of scientific papers, notes, and research and give you the distilled knowledge, no fluff, just the fascinating insights you need to be truly well-informed.
Our mission for you today, the learner, is perhaps one of our most radical yet. We are synthesizing findings from three major new studies that really force a complete recalibration of how we think about biological aging.
We're moving past the simple stuff.
Oh, way past. We're getting beyond the well-worn concepts like just eat less and shifting the focus to these hidden mechanisms, metabolic flux, genetic resilience, and the subtle microbial signals that dictate longevity.
The sources you shared, they paint this incredible interwoven story. I mean, the connection between a pill used by millions for diabetes, the most rigorous dieting regimes, and, unbelievably, the bacteria living in your mouth.
Exactly.
We're going to show you that the body's roadmap to a longer life is just so much more complex than we ever realized.
Yeah, so specifically, we'll cover the revelation that a common drug, metformin, might be acting primarily through the intestine. It's basically running a bacterial food delivery service.
And then we'll get into why dieting works, but maybe not for the reasons we all thought.
Right, and why resilience seems to be a better predictor of lifespan than just, being lean.
And finally, we'll dive into how the tiny microbial communities in your saliva are strongly linked to your biological age and your cognitive health. It's a deep dive into the unexpected levers of longevity, and we're going to give you the gold nuggets. Okay, let's unpack this. Starting with a surprising discovery about metformin, a drug that's been a workhorse for diabetes for, what, decades?
For decades. And for the longest time, the textbook explanation was really straightforward. It was that metformin acts mainly by blocking the liver's ability to churn out glucose, a process called hepatic gluconeogenesis.
So it just slows down the sugar production coming from inside your own body.
That was the canonical view, the universally accepted view. But this new analysis, which used this incredibly precise PT-MRI imaging, it suggests we were missing a fundamental, massive piece of the puzzle.
They found a hidden river of glucose.
A hidden river is a great way to put it. A substantial, previously unrecognized river that is being dramatically enhanced by the drug.
Okay, so what is this river? Where is it flowing?
That's the crucial detail. This river is the flux of glucose moving out of your bloodstream from the circulation.
And into where?
And into the intestinal lumen. It's glucose being actively transferred into the gut itself, past the point of absorption.
And the sheer volume of this is what's so wild. In human subjects with type 2 diabetes on metformin, they found patients excreted, what was it, about 1.65 grams per hour?
1.65 grams per hour of glucose straight into the gut.
So put that into context for us. How much is that really?
Well, to contextualize that for you, that amount is comparable to about 20% of the body's entire glucose output from the liver when you're fasting.
20%. Wow.
This isn't some marginal side effect. This is a major metabolic pathway being manipulated. It really suggests the gut is far more involved in blood sugar control than we ever thought.
But is this just a drug effect? Or is this something that's always happening?
It seems to be a common physiological feature, yeah. Even the individuals not taking metformin were still excreting a noticeable amount. About 0.41 grams per hour.
So metformin just puts its foot on the accelerator.
It puts the foot way down on the accelerator of an existing mechanism.
And they did this striking comparison between species, too.
Yeah, that was fascinating. When they standardized the rate of this glucose transfer by body mass, the efficiency of the process was virtually identical between human controls and mouse controls.
Really? That close?
Almost identical. And metformin boosted this transfer rate by a similar factor in both species, nearly fourfold. That stability across mammals tells you this mechanism is, deeply conserved. It's fundamentally important.
And the imaging was key to figuring out where this was happening.
Oh, absolutely critical. They used a glucose tracer, FDG, and these continuous PT-MRI scans clearly showed the tracer appearing initially in the jejunum.
The first part of the small intestine.
Exactly. So it confirms that this section of the gut is acting as this rapid excretion point for circulating glucose.
So we have this metabolic dump truck, and it's actively depositing precious fuel sugar into the gut. Why? I mean, if the goal is to lower blood sugar, why reroute it there?
That brings us to the microbial payoff.
What?
Once that glucose is in the intestinal lumen, it doesn't just pass through, it immediately becomes food for the vast population of gut bacteria.
So you're basically giving the microbiome a subsidized meal plan.
Exactly. And the researchers confirmed this using stable isotope tracing. They injected C13-labeled glucose into the circulation,
Huh?
and they found that the bacteria metabolized it. They converted the host's glucose into short-chain fatty acids, or SCFAs.
And metformin specifically amplified this process.
Dramatically. The drug significantly increased the incorporation of that C13 tag into propionic acid, which is a key SCFA known for its anti-inflammatory and metabolic benefits.
So what does this all mean then? Metformin is essentially hacking the system.
It is. It's less about directly blocking the liver, though that still happens, and it's much more about driving glucose out of your circulation and into your gut, where it feeds beneficial bacteria.
So this flux directly contributes to the symbiosis between us and our microbiome.
And it acts as a major, previously unrecognized target for lowering blood sugar. It just fundamentally reframes how we look at this drug.
It's not just a regulator anymore. It's a microbiome engineer. It makes sure the good guys get the fuel they need.
That's a perfect way to put it.
Okay, so if metformin is rewriting the rules of the gut, what happens when we look at the oldest, most studied rule in longevity dietary restriction? Because here's what gets really interesting.
Right.
The next set of sources delivers an even bigger paradox about leanness and lifespan.
This takes us to the DiRido study. That's dietary restriction in diversity outbred mice. And this was a massive undertaking. We're talking 960 genetically diverse female mice.
Oh.
And they were tracked across 5 different feeding interventions, everything from eating whenever they want.
The ad libitum group.
Right. All the way to severe caloric restriction.
The basic results, though, they confirmed what we'd expect, right?
For the most part, yes. Lifespan extension was generally proportional to the degree of restriction. The 40% caloric restriction group had the greatest benefit, a median lifespan 36% greater than the control group.
And even the intermittent fasting mice lived longer.
They did. They extended their median lifespans, and they did this with minimal, or in some cases, no net reduction in total calories.
But here's the twist, the paradox.
Yeah.
And this is the gold nugget for you, the learner.
This is the big one. The standard metabolic health markers that everyone chases, reduce adiposity, lower fasting glucose, less body fat, they were surprisingly poor predictors of maximum lifespan within these diet groups.
Wait, I have to stop you there. That makes no sense.
I know.
We're constantly told, culturally and scientifically, that lower body fat and better glucose numbers is always the goal. So how do you explain the fact that weight loss at any age after the diet started was generally associated with a reduced lifespan?
It forces us to distinguish between 2 concepts, metabolic health and biological longevity.
Okay.
Metabolic optimization makes you healthy, sure, but the study argues that lifespan is driven by something else entirely.
Which is
Resilience.
resilience, as in the ability to withstand a shock. How do you even measure that in a mouse?
They quantified it using several measures, but the most powerful longevity indicator they found was something they called phenodelta.
Phenodelta.
This measured the animal's retention of body weight through periods of handling or stress. When researchers handled the mice for measurements, they experienced minor stress. The mouse that lost the least amount of weight during the stress, the mouse that could effectively absorb the shock without degrading, showed the strongest association with a longer lifespan across all the traits they tested.
So the ability to bounce back or just absorb the hit was a better predictor of survival than just being lean and optimized at baseline. That is a powerful reframing.
Absolutely. It suggests the biological systems that confer robust stability are the ones that grant longevity. And this is reinforced by another finding.
What's that?
They observed that higher adiposity in older mice, so beyond 22 months, was generally associated with an increased lifespan.
So having more body fat when you're older is protective.
It seems that retaining energy reserves in late life acts as a vital buffer against mortality.
So you want to be lean early, but having a little reserve, a buffer when you're very old seems to be a good thing. It totally flips the script on chronic caloric depletion.
It really does. And they also delved into blood cell measurements, which were unexpectedly strong predictors.
What kind of signals did they find in the blood?
Well, these hematological traits were highly predictive. Low red blood cell distribution with, or RDW, that really stood out as a key positive predictor.
RDW. That measures the variation in the size of your red blood cells, right?
Exactly. Low RDW suggests a uniform, healthy, stable cellular environment. They also found that a high circulating lymphocyte proportion, indicating a robust immune system, was also positive.
And I remember reading that the intermittent fasting group had a telling metric there.
Yes, the 2-day intermittent fasting group showed a sharply increased RDW by just 10 months of age.
Hmm.
So this suggests that while IF overall extended lifespan, the intervention was causing some kind of physiological disruption, a lack of uniformity in red blood cells, which might be a hidden cost, even if their genetics could compensate for it.
And in the end, the ultimate predictor wasn't diet at all.
It wasn't. While diet accounted for about 7% of the variation in lifespan, genetic background was the largest influence. It explained nearly 24% of the variation.
So longevity isn't something you just earn through restriction.
It's something you are largely built with. They even identified specific genetic markers. For instance, a founder haplotype on chromosome 18 was associated with both a reduced lifespan of about 4 months and also high RDW.
So the key takeaway here is that your internal hardiness, dictated by your genetics and measured by your resilience, matters far more than the exact percentage of calories you cut.
That's it. Longevity is about maximizing your internal hardiness, not just minimizing your intake.
Okay, now let's pivot entirely from the mouse gut and the longevity paradox to the human mouth. The third source provides this fascinating connection between the bacteria we carry in our oral cavity and our biological age and cognitive fitness.
This study focused on a cohort of the oldest old Singaporean adults, a really valuable group with a median chronological age approaching 89 years. And they assessed biological age using the sophisticated DNA methylation clocks, looking for the deviation between a person's calendar age and their estimated biological age.
And the immediate finding was completely counterintuitive. It just flew in the face of what we typically believe about microbiomes.
That's right. I mean, generally, scientists associate high microbial diversity with better health. But here, individuals with a lower biological age deviation, meaning they were biologically younger, they exhibited significantly lower oral microbial diversity.
The opposite of what you'd expect.
The complete opposite.
So the biologically young, very old people didn't have this crowded, diverse jungle of bacteria in their mouths. They had a streamlined, elite team.
Wow.
What was making up this less diverse but seemingly more effective ecosystem?
It stemmed from these compositional shifts involving specialized dominant taxa, specifically short-chain fatty acid-producing genera like Prevotella 7 and Veillonella.
Okay.
They trended toward higher abundance in the biologically younger group. They had this focused operation optimizing for beneficial metabolite production.
And the consequence of this highly specialized, low-diversity ecosystem was measurable differences in brain health.
Yes, the study found a statistically significant link between these specific oral taxa and performance on the Mini Mental State Examination, or MMSE scores, which measure cognitive performance.
So the bacteria in your mouth are talking to your brain.
It seems that way. 2 specific species of Prevotella 7 were positively associated with better MMSE scores. When they compared people at the low end and a high end of abundance for these bacteria, the difference in cognitive scores was clinically relevant, up to a 1.5-point difference.
That is a staggering implication. What's the hypothesized mechanism? How are mouth bacteria talking to the brain?
Well, the hypothesis centers on a metabolite-driven epigenetic pathway.
Okay.
The scFAs produced by these bacteria, likely entering the circulation through the oral mucosa, are acting as epigenetic modifiers. They were linked to methylation changes at specific sites in genes relevant to neural function.
They cited genes like RBFOX1 and P2RY6. What do those do?
RBFOX1 is relevant to neural signaling and has been linked to things like brain amyloidosis, while P2RY6 is implicated in neuroinflammation and synaptic loss.
So the scFAs are basically modulating the expression of these genes.
It seems so. It provides a direct microbial-based link from your oral health to your brain stability and your cognitive youth.
Wow. So whether it's in the gut, being fed by this massive glucose diversion from metformin, or in the mouth promoting cognitive health, the microscopic world seems to be directing our long-term biological trajectory.
And it's all by controlling the flow of these essential metabolites. It reinforces the central theme across all your sources. The host body is in this constant negotiation with its microbial partners.
And maximizing that symbiosis may be the key to aging successfully.
I think so.
So let's bring these 3 segments together for you, the learner. We've seen that the longevity journey is not a simple equation of just restriction. It involves optimizing metabolic fuel flow to beneficial microbial partners, whether that's in the gut via a drug or in the mouth via a specialized ecosystem, and maximizing the host's innate resilience.
Right, that resilience against stress and degradation, the stuff measured by traits like phenodelta.
So this raises a really important question for you to consider. If high adiposity in late life is a positive predictor of lifespan in genetically robust individuals, acting as a vital energy reserve,
And if SCFA-producing bacteria in the mouth are strongly linked to cognitive youth, where should our strategic focus shift?
should we stop striving for maximum leanness and extreme caloric depletion throughout life, and instead focus on cultivating those microbial ecosystems and retaining appropriate energy stores to ensure maximum resilience in the face of inevitable aging and stress?
The true secret to longevity might be about maximizing reserves and optimization, rather than simply pursuing endless restriction and leanness.